Greetings one and all. This is Sandra Balakrishnan. I'll be talking about my research project, that is analysis and evaluation of bioenzymes derived from fruit peels of lemon, orange and pineapple. Bioenzyme was first developed by Dr. Rosukon by using jaggery, kitchen waste and water in the ratio 1:3:10 and fermenting it for a period of 3 months. The liquid that was obtained after fermentation and filtration was known to contain enzymes, alcohol, acetic acid, etc., which justified its use as a multi-purpose liquid used in fertilizing, disinfecting, cleaning, etc. In my study, I had taken bioenzyme solutions made of orange, lemon and pineapple. These peels were added to the separate plastic containers with one part as jaggery and a pinch of yeast to accelerate the fermentation process. Here you can see after a period of 45 days, the fermentation period was over and it was filtered. The pH, odor and the color was noted. You can see that all of them are in the acidic range. The phytochemical screening was done. There saponin, tannin and flavonoids were shown as present. Protein estimation was done here with the help of Lowry's method. Bovine serum albumin was used as a standard. Increasing concentration of bovine serum albumin was treated with the reagents and made into a standard graph. The protein content in each of the enzyme was calculated using the standard graph. The sugar estimation was done with the help of DNSA reagent. Here, the standard was taken as dextrose. Increasing concentration of dextrose was used to make the standard graph, and just like in protein estimation, the concentration of sugar was calculated with the help of standard graph. The acetic acid estimation was done by titrating the diluted solutions of bioenzyme with the help of NaOH. The volume of NaOH used to neutralize the acetic acid present in the sample was taken, and then the acetic acid concentration was found out. The qualitative test for presence of enzymes. Here, the presence of enzymes was carried out indirectly by checking for the presence of its substrate. In the protease test, the biuret was used as an indicator checking for the presence of protein, and BSA was used as a substrate. In the enzyme blank, you can see the biuret has turned the protein into purple color. In the samples that were treated with pineapple, orange and lemon enzyme, you can see that there is no purple coloration, indicating the presence of protease that has broken down the protein. Similarly, in amylase test, starch was used as a substrate and iodine was used as an indicator. In the enzyme blank, you can see that there is dark color indicating the presence of starch. In the samples that were treated with pineapple, orange and lemon bioenzyme, you can see that there is no dark coloration, indicating that the starch was broken down, thereby confirming the presence of amylase. In the lipase test, alkaline full fat milk was taken as a substrate. A few drops of phenolphthalein was added as a pH indicator. In the enzyme blank, you can see that the pH is very high, indicated because of the pink color of the solution. In the samples that were treated with the enzymes, you can see that the lipase present inside the pineapple and orange and lemon bioenzyme had broken down the fat present in the milk and into fatty acid, thereby reducing the pH. Hence, you can see that the color is fading from pink to light pink and eventually white, thereby indicating the presence of lipase. The microbial composition inside the bioenzyme solution was done using spread plate method. The serially diluted solutions were used. In the testing for bacterial colonies, nutrient agar plates were taken and were spread with pineapple, orange and lemon bioenzymes. The agar plates were kept in incubation for 24 hours at 37 degrees Celsius. Here you can see that the orange bioenzyme plate is having a lot of bacterial colonies compared to pineapple and lemon bioenzyme plate. The fungal composition was carried out using YEPD media. Here also 100 microliters of solutions were spread on top of them. The agar plates were kept in incubation for 72 hours at 27 degrees Celsius. Similarly, as in bacterial composition, you can see over here that the orange bioenzyme is showing a lot of microbial fungal growth compared to the pineapple, which has 0 colonies, and lemon, which has only 1 colony. The reason why the orange bioenzyme might contain a lot of microbes is maybe because the acetic acid content present in the orange bioenzyme is less compared to pineapple and lemon bioenzyme. Smear preparation was done from the bacterial colonies and it was subjected to gram staining. Then the slide was kept under 100x power of microscope and checked. You can see that there are different circular coccus bacteria over here and rod-shaped bacteria over here. The bacteria can be tentatively identified as bacillus. In the fungal colonies, you can see that under 40x power of microscope, there is the fungus, that is Aspergillus and Rhizopus, present over here. The microbes present inside the bioenzyme solution can be held responsible for the enzymes and the secondary metabolites. The secondary metabolite, that is saponin, is having foaming ability. So this foaming ability can be exploited in its use as a cleaning agent. The acetic acid content present in the bioenzyme is having antibacterial effect. So that can be again used in disinfecting areas. The flavonoids present in here has antioxidant activities, thereby justifying its use in many different forms, as in sewage treatments, as soil conditioners, etc. Thus, the use of bioenzyme can be a cheap, organic and a healthy alternative to the chemical, synthetic and toxic chemicals that we use in our day-to-day lives. The secondary metabolites and enzymes present in the bioenzyme can be used to degrade harmful and toxic products present in the wastewater into harmless and simple molecules. There has been ample evidence in the use of bioenzyme as soil conditioner because of the good bacteria present in the bioenzyme can be used to increase the yield and thereby increase the fertility of the soil. We know that fermentation increases the good bacterial content present in the food. Thus, bioenzyme can be used also as a probiotic for consumption. Here, the bioenzyme, instead of being made from just from the peels of the fruits, they can be made from the entire fruit. The bioenzyme, which is having different amount of secondary metabolites and enzyme, can help in composting as it accelerates the breakdown of complex organic molecules into simpler and simpler substances. Thus, it can increase the fertility of the soil by making the as a good fertilizer as well, by making the small molecules accessible for the plants. There has been many research papers that shows the antibacterial effects of bioenzymes made from different kitchen waste against different harmful bacteria like Staphylococcus and Streptococcus, etc. Thus, bioenzymes is indeed a living liquid that can be proved as a natural, organic, cheap and eco-friendly alternative to the synthetic, toxic and carcinogenic chemicals that we use in our day-to-day life. These are the references. Thank you.